Hello, and welcome to this roundtable. What are some of the biggest challenges related to the development and delivery of RNA therapeutics? My name is Megan Thomas, and I'm the multimedia editor for DDW and your host for today's roundtable, which is being sponsored by Azenta Life Sciences. Speakers in this roundtable cover the following topics. How has the COVID-19 pandemic influenced the advancement of RNA research? What does the investment landscape look like for RNA therapeutics after COVID? What are some of the biggest challenges related to the delivery of RNA therapeutics? And what does the RNA pipeline look like across disease areas? Our roundtable panellists include Dr. Cruz, Dr. Neill Liptrott, and Reece Armstrong. Welcome, Philip, Neill, and Reece. I'm eager to hear your thoughts on this exciting topic. First, I'll hand over to each of you to introduce yourselves. Philip, would you like to go first?
Thank you very much, Megan. Hello, everyone. It's a pleasure to be here. And my name is Cruz. I'm the Country Medical Director for Moderna in the UK.
Thanks, Philip. Neill, over to you.
Thanks, Megan. Same here. Really pleased to be invited to be involved in this interview webinar. So I'm Neill Liptrott. I'm based at the University of Liverpool in the UK in the Department of Pharmacology and Therapeutics. And I'm also here, I guess, partly representing the Infection Innovation Consortium, which we're a partner, which has been shortened to ICON.
Great. Thank you, Neill. And now over to you, Reece.
Yep. Hi, everyone. My name is Reece Armstrong. I'm the editor for Drug Discovery World. Been working in B2B publishing for about 7-8 years now. So I've worked across a breadth of life sciences titles, from digital health to medical devices, and then eventually pharma. So yeah, really excited to be sat on the panel with Philip and Neill, and excited to hear their thoughts on this interesting area.
Thank you all. I'm eager to start this roundtable discussion now and hear your insights on the topic of RNA therapeutic delivery and development. While some of these questions will be better suited to one or other of you, everyone can feel free to join in and discuss the questions. So, Neill, this first question's for you. What are some of the key considerations that developers need to take into account when thinking about the delivery of RNA therapeutics?
Yeah, so I think that from the delivery perspective, I think most people have realized now that the RNA therapeutics, the application is wide. There's lots of potential applications that could be used for, but I think that the delivery is key. I think because, as we all know, that RNA is relatively fragile and relatively unstable when it's used biologically. So there needs to be a delivery system in order to actually protect it from the body's defenses that will actually degrade it quite quickly. So you need to make sure that it's protected for a sufficient amount of time so they can actually get to where it needs to go. And there's the nanotechnology, sort of liposomes and other delivery platforms that people are investigating. Obviously, liposomes are the ones that are at the forefront at the moment, which obviously Philip will know much better than I will. They've been used in the COVID vaccine programs. Have been shown to be really effective. They can protect the RNAs. They can get them into the muscle cells that are necessary in order to express the RNA into the antigens for the virus. So I think that's really key. I think around the delivery system itself, there are quite a few things I think that need to be considered. I think slightly from my side, because I guess I'm sort of slightly inherently biased because I'm an immunologist by background, is we know that a number of the lipids, particularly the ionizable lipids that are used in the liposomes particularly, while not necessarily sort of overtly cytotoxic or anything, they can cause some inflammation. Now, depending on the application, so say something like a vaccine, that could be really useful. You could have potentially a sort of a novel adjuvant effect there, but it shows that the lipids themselves are not inactive ingredients in these formulations, and there's some debate in the field about how they should be classified from a regulatory perspective. So understanding how those lipids within the formulations can interact with the immune system, I think, is quite key. There's obviously the potential for adverse events, but I think also it's understanding what those lipids and those formulations will do in the application that you're going to use it for. So obviously we think of vaccines probably first and foremost because of the success of the COVID programs. But if we're going to use these to deliver mRNAs, or well, any RNA species really, but we're going to use these to deliver RNAs, but it's going to be an application outside of vaccines, I think that that's something that we definitely need to be sure of. We know that sort of the patisiran, the Onpattro formulation is actually given with sort of an anti-inflammatory beforehand, so we know that some of these can potentially be issues. So understanding those critical components, I think, is key. And we also know that even slight modifications within the lipids, not all of them are the same. So we have to treat them, I think, on a case-by-case basis. On the other side of things, personally, I think that a more targeted approach, depending on the application, I think is going to be quite key. I think these days we know pretty much most of these things, definitely if they go, when they're administered, they'll sort of go into the liver. So liver delivery is maybe not so much of a huge problem. Targeted delivery for vaccines probably isn't a huge issue, because we know that it's an intramuscular injection, they're taken up into the muscle cells, antigen is expressed, and we get the effect that we want. Maybe there's some things around slightly different routes of administration that might have improved that. But I think if we're going to use RNA outside of vaccine programs, then I would definitely say that we need to have an idea of what we need to do to get those RNAs into the appropriate cell. So intracellular delivery, I think, is really key. So are we targeting it to the right organ tissue cell? When it gets into the cell, is it being expressed? Is it able to escape the endosome and actually be expressed into the protein that we're interested in? So I think understanding those principles is really key, right from sort of the early stage of development through to actually, preclinical models and into clinical studies, making sure that you're getting your RNA to where it needs to go and that it's expressed. I think that they're certainly the things that I sort of would consider most important from the program.
Yes, absolutely. Thank you, Neill. Philip, as Neill mentioned, we've seen great progress made in the use of RNA for infectious diseases such as COVID-19. Can these types of successes be translated into other fields such as oncology?
Yes, of course. Thank you very much for the question, Megan. I guess really just to highlight that mRNA vaccines, or even the study around mRNA, did not start during the pandemic. There has been really a groundwork even before that, decade before that, actually. And so it's just really where during the pandemic, a lot of attention and focus and support went into it. And I guess really that helped the technology gain its ground, because all of the scientists really pivoted to focus on solutions against the pandemic. So I guess really let's kind of like leverage on the gains during the pandemic, and of course look at where it originally was intended into, where the study in oncology was for mRNA therapeutics / vaccines. So yes, I mean, really the learnings that we have gained in such an amount of time, when you look at the number of clinical trials, it has greatly multiplied, which is great for in terms of addressing these types of diseases, such as those in oncology and specific type of cancers. But then again, when you look at the number of studies, currently most are in phase 2, and most are targeting solid tumors. And therefore, there is that momentum that has gained. And of course, one key challenge that we need to be mindful, especially dealing with the platform technology, is on not just the durability, but also on the safety profile long term. So there needs to be an ongoing and real-time surveillance of safety and efficacy and effectiveness.
Thank you very much, Philip. That was a great answer. Now, over to you, Reece. What are some of the potentials for RNA that interest you?
Yeah, well, like Neill and Philip have alluded to, there's such a wide use case for RNA therapies at the moment. And obviously, it has been bolstered by COVID-19, and we're starting to see a lot of developers investigate different types of areas, across infectious diseases, oncology. For me recently, sort of being a journalist in the field, I was quite interested in one of the use cases for antibiotics, which we know has become sort of increasingly resistant, and as time goes on is going to represent a major problem for society and life sciences going forward. So there was a clinical study investigated at the Institute for Molecular Infection Biology and the Helmholtz Institute for RNA-based Infection Research, and they're essentially trying to block the genes of bacteria using mRNA to kill the bacterium. So I think that's just one example, which is a really novel use case for mRNA, and sort of highlights quite a unique application for it, really. I mean, obviously, like Philip says, cancer is the big one. It is typically one of the most investigated diseases within pharma for good reason. It represents such a burden on mortality in the healthcare systems, and of course patients. I think any advances will be really exciting, especially if we can remove ourselves from the typical approach to treating cancer, which is, chemotherapy, and that's very, very hard on the patient and not always successful. So yeah, I'd be interested in any sort of oncology developments, and we have Moderna's work with Merck on the personalized cancer vaccines for patients, and I think that had some quite positive findings from the phase 2b keynote clinical trial. And then additionally, just another example was a product being investigated in the field of women's health by the biotech Versameb. And that's being developed for stress urinary incontinence, and that's using mRNA to translate DNA into patients so their own cells can help repair any damage in the body and then protect itself against disease. And that's really important because typically with SUI treatment, we haven't had an approved medicine, so we've only had surgical techniques. And when we know some of the, for example, the sling meshes that have been used in that area have succumbed to a lot of side effects and are quite bad for the patients in many cases. So yeah, just another really sort of unique example of where this technology can be applied to outside of the typical infectious diseases and cancers, really.
And Megan, if I can just acknowledge that, and thank you very much, Reece, for the insight. I'd like to highlight that if I could turn back the time, and because I'm trained in pediatrics, I would have probably subspecialized in immunology. And I will speak that to Neill's specialty, because right now the field of immunology, we're just really truly beginning to understand ourselves, our immune system, how we react, and therefore I think really what is crucial when we talk about mRNA technologies is understanding our immune system, understanding how our T cells work, how they interact. When you talk about, Reece, of the oncology approach, having another treatment such as immune checkpoint inhibitor, realizing that when you want to address cancer, especially realizing that there is a tumor microenvironment there where the immune system is switched off, or there is a disequilibrium of sorts. And that's where we're trying to understand more of ourselves, our human immune responses, and that's where I think is key for us to better propagate this technology forward.
Yes, thank you, Philip, and thank you, Reece, for your answer. It does seem a trend, this multidisciplinary approach. And so, Neill, I wonder if you could weigh in on how beneficial you think collaborative models can be for developers of these therapies.
I think it's probably a slightly cliche of me to say that obviously we need collaboration in development of any therapeutic, really. I think that goes as read. But I think particularly with this kind of technology, it is so multidisciplinary. We think about the RNA therapeutic itself as the payload, if you like, and then we have the delivery system, and then there are potentially other things that we might do to make the delivery system more stable or target it, whatever we might do to decorate it or protect it from the body. And as Philip was saying, also potentially protect it from the immune system to make sure we get the actual response that we want. I think that we need people from all different fields, from sort of chemistry, materials science, immunologists, oncologists, infectious disease specialists. I think we need people all the way through that, particularly with this kind of combination, because if we think about it now, and I completely agree with Philip's point about that, that obviously from the pandemic, what's happened is none of this is necessarily new per se. It's been going around for decades. It's just that the pandemic has actually shown what can happen with this kind of technology, how successful it can be when there is a concerted effort and a multidisciplinary effort to actually get these things into clinic for patient benefit. So I think it's sort of proven itself that way. But I think that you need that collaboration across, sort of right from the bench all the way through into early phase clinical trials, because you've got potentially a delivery system with a payload that might be at slightly different technology readiness levels. You might have the delivery system might be quite well known. If it's a liposome, if you like, they've been around for a long time, but the RNA might be sort of been modified in a particular way, and then you put them together into this final product that is at different combinations at different stages. So I think people who can understand those differences and potentially tease those things out are really key. And being able to take that through from the sort of the bench, if you like, into those early phase clinical trials and beyond, I think you need people who've worked in relevant fields so that they can almost sort of speak each other's language. I don't think anyone is ever sort of a true multidisciplinary scientist. We all naturally fall into the areas we're interested in or the areas that we're really good at. But I think working with people in those other fields is absolutely key. You know, and you see there are different organizations and different institutions where they're trying to do those kind of things. Like, so with an ICOM, we have the human challenge unit where we can do that early phase sort of vaccines assessments on the earlier stage. You know, there's work that we do within the nanotherapeutics platform, and there are other organizations and infrastructures, I think, that are doing similar things where they realize that to actually realize the benefit and the potential of these, everyone really sort of has to start speaking to each other a lot more clearly. So I think that it goes without saying that the sort of collaboration is key to the successful development of any therapeutic. But I definitely think in this field where there is additional layers of complexity, and as Philip was actually saying, you know, so to carry on from one of Philip's points about, we know from vaccines there is the potential hesitancy that some people can have, and I think that we also have to work together as scientists to actually not just get the therapeutic through into the clinic, but to actually dispel some of those myths and show that these things are really effective, that they can be used for all these different applications. So I think the collaboration also goes a little bit beyond the science, if you like, and the clinical side of things. I think there's also sort of a social aspect to it as well. And I think that collaboration with patient groups and, you know, within the different fields that this might be applied to, I think that's absolutely critical. I think sometimes we can sort of neglect to include those kinds of patient groups and advocacy groups within these kind of discussions and trials, and think their voice is something that should definitely be heard loud and clear, as well as the scientists.
Oh, most definitely, and add to that, Megan, if that's okay, because one of the key topics that is really high on the agenda here in the UK, especially in research, is inclusion and diversity. We want to have clinical trials that truly represent our population. However, when we look at the therapeutic side of things, sometimes we have difficulty in recruiting patients. When you talk about rare diseases or those protein-based diseases, it's an N of 1. So again, the challenge there, and leveraging upon the collaboration that Neill mentioned, is how can we work together with regulatory authorities, how to assess, review and approval of these interventions. So that's a key ongoing discussion that we've had, and that's one key piece and success during the pandemic, where we see a streamlined approach, looking and reviewing data as they come in, and, you know, having those reviews and then subsequent authorizations for the product to be used. So that should continue, and just leveraging again and identifying and acknowledging that now we need to move forward, need to have a more inclusive and diverse patient population, and hopefully that is something that can be realized.
Yes, thank you both for that insight. Now, Philip, I wonder if we can get a bit technical here. We know cancer is an exceptionally difficult disease to treat. What are some of the challenges and opportunities of using RNA or mRNA specifically in oncology?
Yeah, specifically in oncology, it's quite a unique area of challenge, because one of the mainstays is to look at what are the antigens or points where your immune system can identify and have an adequate immune response towards that specific tumor, let's say. And so this is what we have identified as what we call neoantigens. There's even hot and cold tumors when you talk about oncology. Hot being where there's an increased level of mutations, that there are these antigens that pop up where you can identify and target. However, there are these cold tumors where they're very much immunosuppressive, they're hidden, they're cold. It's hard to identify and come up with a solution targeting those. But then again, looking at just focusing, let's say, on these hot tumors per se. So you have identified these types of mutations, which you can now target. And that's where using computer algorithms, when there's a biopsy, let's say, of a tumor, you can do some sequencing and identify which antigens are present. And then you can design a vaccine towards that and then give it to the patient. The patient develops an immune response against that specific neoantigen in that tumor. So, for example, there is the study that Reece earlier mentioned, where there were 30 plus neoantigens identified. But the thing here is no two individuals can be assumed to have the same tumor antigen makeup. So it therefore can begin to assume as a personalized cancer vaccine approach. And so this is where the novel approach of having a personalized immune response towards a specific area of a tumor, having that maximum immune response, is a distinct advantage. And so that also comes to mind, how would regulatory authorities review and approve that approach? So that is an ongoing discussion, because you can't really say that one cancer vaccine is similar to the other, because that is, in its essence, it's the personalized approach. And so that is an ongoing discussion. But that's very exciting in the sense that you now have a targeted approach, and therefore that leads to that mRNA platform technology advantage in oncology.
Yes, absolutely. Thank you. Now, Reece, with that sort of regulatory theme in mind, are we seeing increased activity throughout the industry and government in regard to solving RNA delivery challenges?
Yeah, absolutely. I mean, the fact we've got Philip and Neill on the roundtable today speaks for that. But I guess coming from a UK-based perspective, since the pandemic, we've seen the government invest a lot into challenges with RNA and mRNA. So I think, for instance, earlier this year there was a centre for intracellular delivery launched by the UK between Imperial, the Medicines Discovery Catapult, and a few of the universities as well. So a really sort of concerted effort between government, industry, academia, which is always important. And I think that's intended to help drug discovery developers access technologies and capabilities in intracellular drug delivery, which will be really important for these medicines going forward. And I believe, as Neill mentioned earlier, that'll be for lipid nanoparticle formulations as well. So, you know, this technology, which is really at the forefront of this field, really. And yeah, like Neill mentioned earlier as well, the centre will help with some of the foundational challenges for RNA therapy. So things like efficacy, stability, performance, and also immune reactions, which, as we know, are going to be a major consideration for these drugs. So that's really just, you know, one example of the UK's perspective sort of after the pandemic. Early this year we had the BioNTech partnership as well with the UK government, and I think that's for personalized cancer vaccines for up to 10,000 patients by the end of this decade. So yeah, we're seeing a lot of effort in regard from the UK, and I believe we'll have other countries as well following the example and doing their own thing.
Can I just add to that as well, because Reece raised, it's that centre, the Intracellular Drug Delivery Centre that Reece mentioned. We're a partner in that as well, and I think that there's obviously the scientific and the technical aspects that we're trying to support the UK ecosystem in the delivery challenges for it. In the areas that Reece mentions around sort of safety, efficacy, biodistribution, understanding what these things do in the body. But we are also liaising with MHRA on that as well. We're trying to keep an open dialogue with them, and I think that that's key from the regulatory perspective. One of the things that and I imagine Philip will have certainly had these conversations as well with sort of regulatory bodies, is that the field is moving so fast, which is great, absolutely fantastic to see this innovation and see this benefit coming through. But you can imagine, you know, that if the sort of the people at the bench who are developing these things are finding it difficult to keep up with all the challenges and changes in the field, then the regulators, with everything that they've got on their plate as well, you know, they're trying to digest it. And I think that conversation is absolutely critical, not just about getting these therapeutics approved, but trying to have that sort of iterative dialogue with the regulators so that they've got a good idea of sort of what's on the horizon, what's coming, what might the next challenge or the next therapeutic look like. And maybe if there are certain things, alluding to what Philip was saying, you know, this around if it's not necessarily personalized, is it stratified? Could that be a way forward? You know, that you could stratify certain antigens or certain people, so at least then it might be, and this is very much in inverted commas, easier approve something, rather than sort of doing it on an individual basis. But I think this comes back to my point on the question that you asked, Megan, about the collaborative side of things as well. Is that we talk about the scientists, we talk about the patients, and obviously the regulators is sort of there's probably many more legs to that stool, if you like, but that three-legged stool is absolutely critical to keep all those things balanced. And I think that's just trying to keep up with things is difficult enough. But I think hopefully some of these infrastructures with those interactions with the regulatory bodies, as we're trying to do with IDDC, I think is really important. So we can also be pragmatic. I think that's the thing as well. It's we're not going to be able to solve everything straight off, but I think if we can move forward with the most pragmatic approach we can, I think that's going to be really key.
Thank you, Neill. Now, another question here for you. What applications does RNA have in the development of next generation vaccines for infectious diseases?
I don't think it necessarily has to be for infectious diseases, as Philip was saying. I think the technology is potentially so flexible and adaptable. It has a reach into many different applications, as long as it's applied in the most pragmatic way forward. I think from the infectious disease perspective, I think the most straightforward way forward is obviously vaccine development. And we've seen a lot of people that we've worked with who've worked in the traditional vaccine space, if you like, and now companies are starting up programs within the RNA space as well. I think to try and pick up on the flexibility and adaptability of the technology, that potentially if there are breakthrough mutations or variants that come through in different pandemics, theoretically, you've got a platform that can be adapted as the antigens might change. Again, as Philip was saying, very similar in the oncology field. You can adapt to try and make the protein for whatever function that you're particularly interested in. And we know that a lot of companies are looking at that to look at sort of pandemic preparedness, from an infectious disease perspective. So I think that the potential flexibility and adaptability of those applications is where the next generation sort of vaccines would sort of be based, and where they'd be coming from, particularly from emerging infections that maybe we don't really know much about at the moment. Maybe as was done during the COVID pandemic, a frontline initial sort of first response vaccines, while other therapeutics and maybe multivalent vaccines are coming in the background for longer term protection. So I think there's definitely a route there from the infectious diseases space in the vaccine applications. But then there's always that slight dose of cold water, is the fact that obviously there has to be regulatory approval for these things, and how do we potentially approve something that we might have a delivery system that has RNA against a number of different proteins. It might not just be one. That gets really complicated. But I think although it's a problem to be solved, I think it's a good problem to have. The fact that we do have this technology, that we do now have this in our armament against, for pandemic preparedness. I think that's really key. I think that everything else is something that can be understood and can be resolved. Again, I know we keep coming back to it, but I think it was sort of a big push forward for RNA therapeutics. The COVID pandemic has shown what can be done if everyone really sort of focuses on a particular problem. So I think there's definitely a case for it. I think that we know that a lot of companies have, if they haven't already, are starting up programs within the space to explore what they could do for the fields, for the infectious disease fields that they're already producing vaccines for, and seeing how they can potentially pivot some of their work towards that.
And I guess really just to highlight the advantage of mRNA vaccines is the scalability and also the speed in terms of producing an mRNA vaccine. Before, when we would make traditional vaccines, it would take 10 to 15 years. Now really it has shrunk into that expedited timeline. But do note that the balance there is, when you look at production, is also the challenge of storage stability. It has to be in an ultra cold operation. However, I guess really the momentum now is working towards a more stable, should I say, vaccine or product, and that has to do with the length of the strand of mRNA, and it's kind of negatively charged, and therefore part of that looks into the chemistry of it really in terms of shortening it and making it more temperature stable.
Yeah, I think that cold chain is as Philip was saying, is an issue. And I think what we also have to think about is, potentially some of the vaccines that are coming forward are we should always, the inclusivity side of things, again, we should always make sure that these therapeutics are going to be beneficial for people in low and middle income countries. And whereas we can scale and make them fairly straightforwardly in the UK and in sort of in other countries, maybe low and middle income countries might not have that manufacturing capability on site at least. So we need to think about that cold chain responsibility, I think, because we do know that some of the vaccines that are being studied by different companies at the moment, obviously COVID-19, SARS-CoV-2 is a global threat, but when we look at things like dengue virus and Zika, they're relatively localized in areas that might be sort of low and middle income countries. So if we're developing those really interesting, really flexible, advanced next generation vaccines, I think we always have to keep a weather eye on thinking, well, how economically viable will this be to actually deploy locally and deal with those challenges. We might be able to sort of change the sort of fill and finish quite easily sort of in a country like the UK, but that might be more challenging in low and middle income countries. And I think that health economic benefit, as well as the scientific and biological aspects, has to be considered.
Yes, absolutely. And following on from that, perhaps you think this is more of a yes or no question, but do you anticipate that traditional vaccine developers will move towards RNA-based vaccines?
I'd say personally, I don't think that it would ever be sort of a question of putting all your all the eggs in one basket. I think that there's still certainly a place for those more traditional vaccines, I think, but you could sort of see, or in my opinion, you could see companies that are pivoting towards this to explore the technology, to see how it's applicable for diseases that they're more familiar with, that they have a strong track record and experience in. You can definitely see that they're exploring it. And I think if we come back just slightly to the vaccine infectious disease space, there is that option almost sort of like for an immediate response, with these kind of vaccines, and maybe the more traditional ones would be the longer term, protection as sort of pandemics come through. Philip obviously has more experience of this than I do. I can't imagine a company would pivot completely away from what they have done, but I would imagine that they're looking into it. And I think that from my experience and my understanding as well, is on the delivery side of things, obviously people have focused a lot on the non-viral delivery systems because of the success. We know that people are looking at viral vectors as well, but there are issues there around that need to be solved around potential toxicities and problematic with those sort of with those viral delivery systems. But the non-viral, I think, have really sort of proven their worth. And obviously there is a big thing around lipids and liposomes at the moment, but that doesn't mean that there aren't other delivery technologies that are in the pipeline that, as Philip was saying, people have worked on for decades for nucleic acids, for other applications, and that you hope now that this is sort of giving this whole field sort of a big nudge forward to show that the two, pardon the pun, go hand in hand, the delivery system and the RNA.
Totally. I imagine a world of coexistence. And really what we're seeing, and this is the pediatrician in me, I feel an emotional attachment to the traditional vaccines, and especially in the childhood vaccination immunization space, there definitely is a role there. Plus you have the heritage of a favorable safety risk benefit profile that has been gained through decades and decades of usage. And so you're already comfortable with this vaccine technology that has been there. And so I think it's more complementary. I mean, companies are looking, as Neill mentioned, they're exploring it as part, could be, their way forward, but there still has a role for the traditional vaccines.
Great, thank you both. Reece, a question for you here. What are some of the clinical trials using RNA that have caught your attention recently?
Oh God, well, there's been quite a lot, but yeah, I guess coming from this year, there was a study on pancreatic cancer by scientists at the Memorial Sloan Kettering Cancer Center. It was a smaller study at first, and it basically showed it was a mRNA type of vaccine, which was showed to delay or prevent relapses in around 8 patients. So small scale numbers, but it's now moved on to a phase 2 clinical trial to expand that, and hopefully, if we'll see some encouraging results from a larger cohort. We know pancreatic cancer's typically it's one of the most common, it's one of the hardest to treat, and unfortunately has one of the highest mortality rates as well. So yeah, it's like a point I was going back to earlier. If we can see some big advances in fields such as pancreatic cancer, then it'll be really exciting. And that's it's a similar thing which we saw with the COVID vaccines, which the mRNA vaccines for cancer, custom made for every person, and use proteins which are found on the pancreatic tumors, called neoantigens, which Philip alluded to earlier, and it's meant to stimulate an immune response, so the body recognises that the cells are dangerous, and so it can protect itself. There was also a study from Cartesian Therapeutics, and they're combining RNA and CAR T therapy for patients with a autoimmune neurological disorder, MG. And again, that was only a small scale clinical trial, I think in 14 patients. But it showed long-lasting clinical improvements for those patients, and it was also the first clinical trial to combine the use of CAR T engineered alongside RNA to treat that autoimmune disorder. And it just shows that despite research going back 20 years in this area, a little bit more, there's still a lot of innovation to be discovered in regard to RNA. So yeah, those were just two that sort of stood out to me, but obviously there's so much activity in this space. So you could list trial after trial, but yeah, those were two quite exciting ones, I thought.
Thank you, Reece. Now, Philip, following on from what has been said about some of the challenges of delivering RNA or mRNA therapies for cancers, how does this contrast between solid and liquid tumours?
Yeah, exactly. And I guess really where what I mentioned about the concept of hot and cold would be, but when you talk about solid and liquid tumors, crucial to that is the manner of delivery of the therapeutic. And so that's where the work is pouring out and the number of research is there. But also I'd like to highlight that a crucial step is understanding really our immune system. Again, leading up to the value of knowing your immunology. But then like how can we instruct, because it's all about the mRNA instructing the body. So it's your body doing the intervention. It's not this vaccine. The vaccine, what it does is provides an instruction, and then your immune system does the rest. And so that talks about really the natural way of us understanding how nature works and leveraging upon that understanding. But then again, a key piece there is knowing more. For example, your body is tricked by these tumor cells to create an environment that the immune system is suppressed. So if you have an intervention that leverages upon your immune system, then therefore the response would be suboptimal because it's not working well in the first instance. And I would also throw in the concept of T-cell exhaustion, where your immune cells are put to the brink of exhaustion by these tumor signals. And therefore, really, the more important ways is really to understand how to overcome these situations in that, what I say, tumor microenvironment. And that's where I think really the understanding and the research needs to be poured into, where all the efforts to clearly define how can we navigate around these challenges of less optimal T-cell function. So you have these combinations, not just an mRNA vaccine, but an established modality as well. So in combination, they could exert the best method of treatment. So that's where I think really this is focusing upon at the moment.
Thank you very much. So this next question I have is for everyone, which in some ways has been answered already. It's, how has the COVID-19 pandemic influenced the advancement of RNA research? Again, this has been discussed, but it would be great to get everyone's insight, maybe a lesson learned from this period. Neill, can we start with you?
Sure, yeah. I think so. I think, as Philip definitely said at the top of the call, and I think as you say, we've mentioned a couple of times, the the applications themselves aren't, or the technology itself is not new. I think it's important to acknowledge the fact that this has been around for a long time. It's not something that sort of sprung up out of nowhere during the pandemic. I think definitely what the pandemic showed was the direct applicability of the technology to a disease. It just in this instance, obviously it was a pandemic, it was infectious disease rather than something else. I think it showed what it can do. I think it really showed, if you have multidisciplinary teams and infrastructures that are all focused on one particular task, what can be done, and that's right across from the bench, preclinical trials, regulatory bodies, everything. If everyone is focused on the same thing, it shows that what we can sort of do with this technology. So I think that it's certainly shone a light on the technology that people have been working on for a long time. So I think that's really good. I think it's also shown the adaptability of the technology. One thing I would say though is, and to touch on what Philip was saying, and I agree completely, is that it is a fantastic technology. We have to be realistic in how we apply it. It's not necessarily a universal panacea that we can now apply to every single disease. As Philip was saying there from the oncology space, it might be just because you can make a particular antigen doesn't mean it can be applied in that particular form of cancer. There are lots of obviously sort of caveats around that, but I think one could say that about any therapeutic really. I think that we have to keep the optics as pragmatic as possible and say that what we now have is, I mean, obviously it's been worked on for a long time, but what we could say at this point now, we've got a demonstrably proven technology, and now it's about exploring how it can find its place in different disease applications. Whether, as I say, it's not a universal panacea. Where can it be deployed? Can it be deployed synergistically with another therapeutic, as Philip was saying? And that's not just within oncology; that's within anything. And I think that's where we sort of are now. I think it's quite exciting to be at this stage. It's been proven. So how can it be applied, and how can we apply that sort of realistically? I think is the most important thing. But I think also to say, as well as the actual clinical application, it's a very exciting technology to be able to deploy to understand biology. I'm glad that Philip always comes back to immunology, because obviously I'm biased, but I think as well, understanding disease processes in general, I think it's also a useful tool as well as a therapeutic. And I think that people have forgotten that a little bit, really, that we can actually, as well as using it to treat some of these conditions, we can also use it to try and understand some of these conditions as well. So I think having the light shone on it, I think, sort of being pushed back to the forefront after decades of research, I think, and sort of accelerating people's work and sort of view that they've been working on. I think that's for me is one of the big things we can take away from it.
Yes, thank you. And Philip, what do you think?
Well, I would say that together we can do more, and that's somehow exhibited by the pandemic, where all the support went into research against SARS-CoV-2, where everyone was on that. But I want to acknowledge, though, we're now living in an era where it's science is judged on preprint publications. You ride a cab, and then the cab driver would be knowledgeable about PCR. They know what PCR is. They know about antigens. And so just on that piece, I like to make an appeal to make mRNA as inclusive as possible, especially around education, and to especially, well, we're working on medical education, especially on mRNA technology towards healthcare professionals, because we can never leave them out in terms of the understanding. And it's been years since they graduated from medical school, and therefore a refresh of their understanding of what the mRNA is, because once mRNA vaccine or a medicine or a therapeutic comes on board, there needs to be that acceptance as well from the healthcare professional community. And therefore, we need to start now. Arm them with the basics and answer queries from them, because of course they'll be very inquisitive about what the technology is. It's something novel. It's something that they need to understand for them to be better advocates and for their patients in the future. So the time is now.
Yes, absolutely. Thank you. Reece, how about you?
Yeah, I mean, there's a couple of points I can pick up there from Neill and Philip. I think what was amazing during the pandemic, which is being said a lot, was just the speed at which research progressed, and that was thanks to the collaborative aspect across industry, academia, and government. Really, when you think about it, that expedited process was remarkable, really, to get those vaccines to market so quickly. So I think just seeing the success there in regards to the mRNA platforms, the RNA platforms, will have encouraged a lot of developers to explore that field, or if they have a disease which they think is applicable to that platform, then to go down that route, because they've seen it work. It's an adage in pharma that once success happens, more companies will want to chase that success. So you'll get that type of effort. And I think the medical education point is really interesting as well, but from a patient's perspective as well, the era we live in now, it can be one of skepticism, and we saw that throughout the pandemic. So to have an effort from the medical community to really get information out there onto how vaccines work and how RNA and mRNA works within the body and why it's important and what it can do for diseases as well, is really important, just to counteract any type of skepticism you might have on patients' behalf, which can it can be well founded. There's been issues within the clinical trial community going back decades where unfortunately patients have died, and anyone looking at those situations might feel an air of skepticism about science and medicine. But to have the education there from all aspects of industry, I think is really important, especially when you're talking about innovative medicines such as RNA.
Yes, thank you. And thank you, everyone. This has been a really interesting roundtable. Before we head on to the Q&A portion, I'd just like to remind everyone watching that this roundtable will be available to access on demand on the DDW website, www.ddw-online.com. Please also submit questions for our panel in the chat feature. Now, before we head on to the Q&A, we will hear from our sponsors, Azenta Life Sciences.
Thank you so much for joining us for this roundtable discussion. I'd like to get into a few more details about how Azenta can help you with your mRNA synthesis and sequencing for precision therapeutics. When we're thinking about the mRNA therapeutics workflow as a whole, we are thinking about the unique and precise synthesis that's required, as well as that sequencing to confirm that the product is correct. And it's really critical to do this early and often to make sure that everything is working correctly. When we're thinking about that discovery workflow, after we've identified our mechanism for the disease and identified our treatment target or potential biomarkers, we're going to actually create that RNA therapeutic. This is the first critical step. We might create one, we might create several different therapies in order for us to test and determine which one's going to be correct. Now, as we go through these later developmental stages, we're going to start adjusting dosages, delivery methods. We're going to start doing those clinical studies and clinical trials. But during this entire process, we really want to make sure that we're maintaining fidelity across the board, especially as we start doing different iterations of our product. And so in that first stage, when we're creating that therapeutic, we really want to think about high quality synthesis solutions. At Azenta, the workflow goes as follows. We're going to start with that DNA sequence again, whichever area it might be, and we're going to create our identified product of interest. That's going to include cap and poly A tail with a very specific poly A tail length. Once we've identified that area and synthesized it, we're going to clone it and do Sanger sequencing to ensure that that plasmid is totally correct. And when we're thinking about that, we can think of not just the insert itself, but also that backbone to make sure that there's no errors in the backbone, that we're getting that high fidelity for the entire product. Once we have that perfect plasmid, we're going to go ahead and prep the plasmid, linearize it, and then do in vitro transcription. Now, one thing that's unique at Azenta is that we do offer both linear or circular mRNA, depending upon the product of choice that you prefer. And then the last step, still very important though, is purification and quality control. And that's going to give us to this end product. Now, I have this as just one simple arrow, purification and quality control. but it's actually a fairly complex process unto itself. At Azenta, we offer some novel sequencing solutions to really ensure that that product that you're getting is exactly what you want. When we're thinking about sequencing that mRNA, we want to get that full length all in one fell swoop, which means we're thinking of long read PacBio sequencing. Now, traditional PacBio sequencing methods on mRNA are going to involve using a template switching reverse transcription approach, typically with a poly-DT. We're going to attach to that poly A tail and read that whole product. And this is all well and good. However, when we're looking at that method, one thing to consider is that that poly T can attach anywhere within the poly A, so we're not necessarily going right on the end, and so we don't know for sure the exact length of the poly A tail. And all of this sequencing is contingent upon a poly A tail being present, which means we're not going to be able to identify any products that are not polyadenylated. Again, really critical if we're in our earlier developmental stages and we want to ensure that we're getting that correct product, that we're not having any byproducts or partial products. At Azenta, we've created a proprietary workflow that involves a 3' adapter ligation that attaches onto the end of the product. The poly A tail is not required for this approach, so we can actually attach onto any and all products that are present, and then use that template switching RT to finish the PacBio sequencing product. This means that we can determine the exact poly A tail length. Really, really critical because the poly A tail length has impacts in both degradation of product, how long the product actually stays in the cell, as well as translation. All of these things impacted by poly A. So we really want to make sure that that poly A tail is exactly correct. We're also going to be able to determine if there's any byproducts that are present, short, long, complex products, and really importantly too, when you're thinking about your later, more mature product, when you've already done that screening and you're starting to do dosages, delivery methods, you might have a bivalent or trivalent mRNA vaccine or therapeutic. We can actually determine ratios of products. So especially when you're working on that blend and you want to make sure that the blend ratio is correct and you don't have anything else present, you're going to be able to see all of that with this sequencing solution. So at Azenta, using both our synthesis and novel sequencing solutions, we really offer a fully streamlined approach to bring you your RNA therapeutics to market faster. I invite you to check out our website if you have any questions or would like to learn more. And of course, feel free to reach out to us. And we thank you so much for joining us at this roundtable discussion today.
Now, onto the Q&A. Right, I think everyone will be able to weigh in on this one. What does the investment landscape look like for RNA therapeutics after COVID? Philip, given Moderna's involvement in the journey to a COVID vaccine, can we start with you?
Yes, without giving specifics, there's a huge amount of investment going into this, not just financial investment, but scientific research investment as well, because we see this as a possible modality of the future, and therefore investment really is the lifeblood of how this technology could be pushed forward.
Thanks, Philip. Neill, what about you?
Well, Philip is much more qualified to sort of comment on than I do. What I can say from our perspective is with a bit more of a focus on the work we do around delivery technologies beyond liposomes as well. We work in lots of different delivery technologies. We've certainly been approached by a number of SMEs who are working in the RNA field themselves. They've been working in sort of nucleic acids for a long period of time for various reasons, and now they're looking at seeing whether or not their technology could be used for different biomedical applications in concert with the delivery system. So I think as well as sort of the large pharmaceutical companies, I think that there is a lot of horizon scanning from sort of smaller companies who realize that actually they've got something that they can offer. It might be a very specific technology. It might be something that they can use to support someone else's work. And I think that we've definitely seen that a lot. I think they're realizing that they can apply some of their expertise and technologies in this field. So I can't speak to the actual economics themselves. What I can say is that people we've been aware of for a while are now approaching us to say, what can we do with this effectively? How can we use it? And I think maybe that comes back again. We've sort of gone around quite a bit, and sort of the educational side of things. It's educating everyone across the field, not just the public not just the healthcare professionals, but also potentially some consultancy and sort of tech transfer and support with sort of smaller companies that might be able to contribute something significant to the field as well.
Yes, thank you. And Reece, what do you think?
Yeah, just to jump on Neill's point, really, I think this is a field in which collaboration is inevitable, and we are going to see a lot of probably deal making within this field, especially as the drug delivery system is so important alongside the therapeutics. So, and that's where a lot of the expertise will be wanted from certain companies wanting to collaborate with biotechs or platform providers, so they can really sort of get the best of both worlds in that sense. I think just going back to my example earlier, the UK is investing in this area as well, so that's a strong indicator of investment is here to stay. It's going to be around long term, hopefully. I think the Canadian government is doing a similar thing with a research grant as well. Specialised in the development and delivery of genomics-based RNA therapeutics. So yeah, those examples sort of good indicators that investment should be strong. Within the private sector, smaller companies might have to fight to get those research grants, but yeah, no, it's definitely positive. I think it's a good time to be working within this field.
Thanks, everyone. The next question I have for you here is, what regulatory milestones do you hope to see that will improve the delivery of RNA therapeutics? Philip?
Well, part of the investment should also go into our regulatory framework, because currently our regulatory authorities are under-resourced, and therefore, part of that investment should go into that. What I would envision is really a clear regulatory pathway towards review and approval of these modalities, especially on mRNA technologies, be it a vaccine or a therapeutic, and therefore this huge amount of work will rely on ongoing discussions, and that approach again needs to be in a way that tailor fits the different types of let's say vaccines, be it this mRNA for this type of disease, and therefore that discussion needs to continue.
Yes, thanks, Philip. Neill, do you see any regulatory milestones you think need to be achieved?
I think it comes back to a few things that we've alluded to on the call. I think that trying to keep pace with the huge advancements in the field that people are making, both in the RNA technology itself and the delivery technologies, and then the combinations, trying to understand what people are really trying to get through into the clinic and what needs to be understood from the developers side of things as well. What would the regulators kind of expect to see? Not necessarily an a la carte checklist of things that need to be accomplished to get them through into clinical trials, but that dialogue I think is going to be really important. I think also understanding what we would sort of class as actives and non-actives in these things, as we were saying about the lipids. We know that some of them are not as inactive as we would normally sort of expect them to be, and how those would be approved as sort of by a regulatory body and what they would sort of perceive them as, I think is really key. But I think that I don't think, not to be too reductive, but I don't think that they're challenges that are necessarily specific to this field. I think that it boils down to understanding the technology that you're trying to improve, how it would be used, and again, where it would be used. Is it something sort of a more general approval, or is it an approval for something in a very specific application? Because as Philip was saying earlier, there's going to be differences in how these things are applied and whether they'll work or not. So I think definitely maybe the biggest hurdle, I would say, that and Philip said it himself, is the support and the funding for the regulatory agencies themselves, so that they can be more responsive for these sort of huge changes and adaptations that are coming in the field. I think that's where I would like to see a lot of the support go personally.
Thank you. Reece, where do you think we'll see the biggest advances when it comes to RNA therapies?
As we basically discussed on this call, the real potential of RNA therapies comes in the form that they offer solutions to diseases which have previously been classed as undruggable. So if we think about small molecule therapies and antibody drugs, I think these medicines, they target less than 1% of the human genome. So for diseases such as cancer, which are cellular and biological mutated cells, for instance, we need drugs that can really tackle them at the root cause, or understand, like Philip has mentioned, the immune system properly. And so the fact that RNA specifically target proteins, transcripts, and genes, this increases the amount of druggable targets and should give developers much more options into what they can target. Whether it's infectious diseases, oncology, rare diseases, it just broadens the scope of modalities that developers can hopefully effectively target and bring treatments for conditions we haven't had in the past. So I think that's a real opportunity that these drugs do represent. I can't say, this is a disease it's going to be huge in, but the fact that it has such a widespread is really exciting.
Thanks, Reece. This next question here looks like a good fit for Philip and for Neill. If you think about your expectations for the future of RNA therapeutics 10 years ago, do they align with the landscape today? Philip, can we start with you?
Yes, 10 years ago. So without the pandemic, it really would think that, this would take really some time. And the pandemic happened, and the rest is history, as they say. However, I think really what we would have a view going forward is what we had during the pandemic as a view: cautious optimism. We're optimistic, but at the same time cautious, very methodical, and we just need to be mindful of that.
Thanks, Philip. Neill, what about you?
I would just echo what Philip was saying. Really, I think it's obviously very difficult to sort of be retrospective on it now, with the lens of the pandemic. But I think that there was always that steady build towards the application of this technology. I think that it was probably never going to be a question of if, more a question of when. But then obviously the pandemic accelerated a lot of those things that were needed to get these things through into clinical trials. So I think that that's, if we would take anything from it, I think that's shown that it's pushed everything forward and sort of back into the forefront of people's minds, I would say. So yeah, I think it's the acceleration of these things through into the clinic rather than if,
Thank you. Now, another for you, Neill. In your experience, particularly with ICON, what strategies does a company need in order to stay competitive in the RNA therapeutics market? What links the stool legs together?
I think it's coming back to what I sort of said earlier. I think really it is this sort of holistic approach. I think being able to access infrastructures where they have as much as possible a bench to bedside approach. I think that if there are multiple different providers that can be a bit disconnected, I think if people have a track record of working together, they have sort of harmonized approaches to the way that they do things, and that there's an almost seamless integration of the steps as you move from the bench to the bedside. I think that's a huge support. And so obviously there are infrastructures in infectious disease like ICON, which is fantastic. You can go from the bench where we're sort of supporting people in the nanotherapeutic space on the delivery technology itself, through to things like the human challenge model. Outside of infectious diseases specifically, and Reece mentioned it before, there's the Intracellular Drug Delivery Center that we established in January this year, which is more agnostic on the applications of the delivery system and the RNA technology. So it doesn't have a particular focus area anywhere, but within that space, we're trying to support the ecosystem, I guess, a little bit more sort of towards the left of the development pipeline, trying to understand the different components of the delivery systems, so that then we can then work with infrastructures like ICON if there is an application in infectious disease, or if there's an application in something else. I think if we're a bit agnostic, then we can see where the technology can go, and then it's up to the experts in their specific fields and applications to take it and run with it, if you like. But I think having that pretty seamless bench to bedside approach, I think, and having sort of companies, whether it's SMEs, large pharma, whoever it might be, who can access those kind of infrastructures, I think is really key. That joined up thinking, I think, is really vital to. Obviously, we're talking about RNA therapeutics here today, but I think any therapeutic development is really, really important to translate it.
Thank you. Now, Philip, this is probably a good one for you. How do you perceive the ethical considerations specific to the development of RNA therapeutics? Are they limiting or enabling for the field?
Well, there's no really difference of how we are currently looking at clinical trials per se. So there's that level of vigilance that goes into a clinical trial, which we need to uphold when we do the mRNA clinical trials as well. And so there would be also a board who looks at safety and primarily on the patients involved in clinical trials. But then again, like I said, we need to maintain the pharmacovigilance, the monitoring throughout, beyond the clinical trials. And so this is alluding to real world evidence, monitoring, working closely with regulatory authorities, keeping a close watch. And then the infrastructure is there on safety and reporting. And therefore, this is a responsibility not just for the physician, but everyone as well, that if you have any symptom that you think is due to a treatment or a modality, just go ahead and report and let your health professional know.
Thanks, Philip. A final question here. Neill, what role do you expect your company to play in the advancement of RNA-based treatments over the next 5 to 10 years?
Wow, that is a big question. So I think what I can say is I'd like to think that at the university we've made quite a good head start on that already. So you can see that, organizations in general in the field, I think, but academic organizations are realizing that these therapeutics are here and they're here to stay. And so now greater support for the sort of investigation, those things I think is really key. An example of that is ICON. So we joined ICON as a platform within that organization, I think early 21, because they realized that these kind of therapeutics were here and they're going to increasingly be used, particularly in the infectious disease space, where ICON is uniquely situated for vaccine applications. So we are working with companies that can come in at different stages within that platform, I think is quite key. To see how ICON, it's obviously been successful, hugely successful already, but to see how that develops and enlarges within that therapeutic space there around these more sort of next generation vaccines, I think that's going to be really exciting to see as Philip was saying, as more companies pivot some of their funding or change some of their streams of how they develop these technologies for maybe the next pandemic, whatever it might be, or that maybe infections in low and middle income countries, which is something that ICON is really keen to support. You can see that having that base there, that sort of expertise and background that we have, that we can bring to ICON, I think is really key. Coming back to the more agnostic side of things, where we think about the intracellular drug delivery center that we're part of, because that is agnostic, I think that's quite key to see that both from the scientific side of things, but also from the UK government perspective, they're realizing the impact of these kind of therapeutics as well, and trying to establish these national infrastructures so that people have either access to the scientific expertise, the analytical capabilities, whatever it might be, or just being able to speak to people in the space and just ask some questions, I think is absolutely critical. So I think we're quite well placed, I think, for the next sort of 5 to 10 years as we start to see sort of a real blossom in how these technologies are going to be applied outside of infectious disease. I think that we're quite well placed for that, and we want to work with companies that are developing these technologies to help them get them through. So I think we're doing quite well in the UK at the moment with these kind of large infrastructures. It shows that people are quite forward thinking quite a bit.
Thanks, Neill. It'll be good to see that unfold over the next 5 to 10 years. And Philip, same question for you. What role do you expect Moderna to play in the advancement of RNA-based treatments over the next 5 to 10 years?
Yes, the role of scientific research really is to pursue mRNA as a platform technology, exploring other uses for mRNA, be it for infectious diseases, oncology, even rare diseases. So that's where the future lies for Moderna.
Great, thank you. That brings us to the end of this roundtable discussion. I'd like to thank everyone for tuning in, and a big thanks to our panel of experts. Please note we will reach out after the event if we were not able to get to your question today. Thank you to the sponsor of today's webinar, Azenta. Thank you, Philip, Neill, and Reece, and thank you again for listening.